neurons, and neuroglia. The neuron is the most basic unit of the nervous system. It's the functional cell of the nervous system. Features of the neuron that are characteristic of them are that they are excitable, so they're capable of electric stimulation. It's not an exaggeration or euphemism either to call this electric. This shifting of ions is effectively what's going on in a battery. In the case of a battery, you're shifting around stuff that's a little smaller, but you still have an imbalance of ions across a semi-permeable membrane. You put it in a situation in which it should carry a charge and move those ions around, and they do. Your nervous system does the exact same thing. Neurons are amitotic. Let's break down that word. A, like not, and mitotic, cell division. Neurons do not divide to make more neurons after you've divided, or after you've been born anyway, or grown up. They are, however, capable of some growth and reorganization. When shortly after you're born, you have all the neurons you'll have your whole life. They just make new connections as you get older, and they get bigger, physically bigger as you get bigger too. If you injure a neuron, it may not die. You may still have the cell body of the neuron alive, and that may regrow some of its axon, or it may rearrange what it connects to best serve its original function. They're also extremely long-lived, provided it has enough nutrition, enough oxygen, not too much of anything that would potentially damage it, not a whole lot of inflammation, free radicals, etc. Your neurons, ideally, all should live as long as you do. The major round, like traditional cell-looking part of the neuron is the cell body, otherwise known as the soma. You'll sometimes also see this referred to as perikaryon. Soma is the most used term, and it's the term I will typically go for. This is the receptive region of a motor neuron. There are several different kinds of neurons. The basic example that we're going to be leaning on most is the motor neuron. This has a pretty clean unidirectional flow of information and a lot of obvious, discrete, different parts to it. So it's a good learning tool. It also is the neuron that's going to stimulate skeletal muscles, which you just learned so much about in the last unit. So this is the receptive region of the neuron. It contains a lot of your standard organelles here. It does your usual cell stuff here. You've got a lot of mitochondria, you've got endoplasmic reticulum, you've got a Golgi apparatus, you've got ribosomes. All the usual stuff lives here, right? You bring things in here, you endocytose things here, you exocytose things here, you generate energy here, you make proteins here, etc. This is where the nucleus is, that's where all your chromosomes are, or your chromatin is. Neurons have very extensive rough endoplasmic reticulum. It turns out that a great deal of your neurotransmitters are little proteins. These are going to need to be transmitted to the ends of your neurons at the very, very far end, and you need to be able to make them to keep up with whatever you've secreted. So it's important to have a lot of rough ER. You have a very extensive Golgi apparatus for packaging and export, and lots of ribosomes, again, for protein making. The rough ER are also known in neurons are also known as Nissl bodies and also known as chromatophilic substance. You're going to see the term Nissl bodies most often. There's a type of microscopic stain called a Nissl stain made up by, I don't know, it's probably some dude named Nissl. And the rough ER takes up this dye really enthusiastically. It turns out to take up this dye very darkly, and so it's named after that stain in a neuron. Clusters of cell bodies within the central nervous system are called nuclei. So if you're talking about the brain, for example, and I say blah, 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 so the nucleus, blah, 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 blah. I'm not talking about the nucleus on the inside of the cell. I'm talking about a cluster of cell bodies within the brain that all are going to talk to one another, process information in a similar way, and send out information in a sort of similar way. Similarly, if we're talking about the same kind of cluster, but it's in the peripheral nervous system, this is referred to as ganglia, or ganglion for singular. Cluster of cell bodies in the peripheral nervous system is a ganglion. I mentioned on the last slide that the soma is the main receptive end of the cell, of the neuron. More specifically, though, the branching appendages of the cell body, the dendrites, are what's really going to bring in that information. The information will be consolidated in the cell body and then zipped down the axon, if it's enough information that it gets. But the dendrites are really the receiving spindles, highly branching receptive spindles that reach away from the cell body and pick up that information. Some of the organelles from the soma are also here, but not nearly as much. You have a very well-suited cytoskeletal architecture inside of each of these dendrites as well. And these are going to receive receptive input from other entities and then transmit it into the cell body. So at any given one of these points on the ends here, at any given one of those points, you'll have another neuron that communicates with it. At the end of this point here, you'll have another neuron with one dendrite that communicates with it, and so forth and so on. A single neuron can make thousands and thousands of connections with other neurons because each one of these points can talk to at least one other neuron, sometimes more. The impulses that are transmitted and the information that's transmitted into the cell body by way of these dendrites are usually what are known as graded potentials. Graded potentials are like small transient impulses of information or of energy that are going to come into the cell. An action potential, as we talked about in the skeletal muscle unit, is an all or nothing electrical impulse. That's going to be generated by an accumulation of graded potentials. If you have enough of those small transient zaps coming into the cell body can build them up into one great big zap and send that information down the axon. The axon is the tail-looking part of the neuron, so down here, this long tail guy. Every neuron has at least one axon. They may branch, and they can be extremely long. You have neurons whose cell bodies are in your brain that reach all the way down your spinal cord and then intersect with something else, and then it intersects with synapses with another neuron whose cell body is in your spinal cord whose axon goes all the way down your leg to the tip of your toe. They can be very, very, very long. A cool thing about action potentials is they are all or nothing signals, and over distances that we have to work with in the body, the signal does not decay with distance, with myelinated axons anyway, and we'll get into the details of that later. But if you get an impulse into your spinal cord, into that neuron, you'll get the same amount of an impulse registering at your toe. It doesn't decay, the information, the energy stays the same. The axon arises from the cell body, from the soma, at an area called the axon hillock. So this is your soma. Each of all of these branchy guys and all of its little mini branches are your dendrites. This is very good artistic rendition, right? So here's your soma, and this part right here that tapers down into the axon is the axon hillock. The axon has some organelles, but it lacks rough endoplasmic reticulum and it lacks Golgi apparatus. These are pretty big wavy structures They have to be situated near the nucleus to really be any good. This is pretty far away from the nucleus, and also it's a skinny, long protuberance that kind of can't fit this sort of thing. So the axon does not have rough ER, Nissl bodies, or the Golgi apparatus. Vesicles made by the Golgi apparatus or made by ribosomes, processed in the rough ER with the ribosomes that are studying the ER there, and then made into vesicles with the Golgi apparatus, will get sent down through cytoskeletal architecture the length of the axon. The axon's filled with microtubules that can help renew the axolemma or shuttle neurotransmitters toward the axon terminal. Remember what the cell membrane of a skeletal muscle was called, of a skeletal muscle fiber was called? It has its own special name, right? The sarcolemma. That should be a hint to you that the axolemma is the plasma membrane of the axon. So those help renew and support the axolemma, and they also help bring those vesicles, processed and exported by the Golgi apparatus, filled with neurotransmitters, down the length of the axon, so they get down here. If up here with the dendrites is the receptive end, this down here must be the transmissive end. Input, output. The axon can also bring organelles and signaling molecules from itself, from the axon terminals, to the cell body for recycling. So sometimes you have some spent stuff down here that you can't really use anymore. You can bring it back up to the cell body by way of the axon for recycling or processing or export or whatever it is you have to do with it. And the movement through here is entirely fueled by ATP. None of this is passive. You have little molecular machines that help move these vesicles along the tracks of the microtubules. Whenever you're doing something like moving big things around, you're going to need energy to do it. So ATP is your currency. Each axon profusely branches at its end. So right here, you can see that super branchy, like any given one of those dendrites. And it can branch so many times, and each one of those can touch another dendrite on another cell. You might have as many as 10,000 branches at the end of your axon. At the axon terminal, you may have 10,000 endings on one neuron. It branches so extensively. You can see, hopefully, between the branching of the dendrites and the branching of the axon terminals, that each neuron is capable of interacting with lots and lots and lots of other neurons. And because of this branching, you don't need, relatively speaking, that many neurons to make enough connections for pretty sophisticated activity. A long axon is referred to as a nerve fiber. Bundles of axons in the central nervous system are referred to as tracts. Bundles of axons when you're in the peripheral nervous system are called nerves. So when we talk about the such and such nerve, you'll know that that represents a bundle of axons that are all going to the same sort of place. In the central nervous system, you'll hear a lot about tracts where, particularly in the brain, where you have a lot of white matter, those are just myelinated axons going from one part of the brain to another part of the brain, or a lot of it is going from one part of the brain to another part of the brain. When you have series of those axons that are all kind of going in similar directions, those are going to have discrete names of different tracts. Physiologically, the axon is the conducting region of the neuron. It's where your electrical impulse is really going to be fully stimulated or fully created and transmitted. So it generates these nerve impulses. It transmits them away from the cell body along the axolemma, the axon plasma membrane. The axon terminals will receive these impulses and release neurotransmitters. Each of these nerve endings, each one of those axon terminals, has a specific region from which it will release its neurotransmitter. That's referred to as the secretory region. These neurotransmitters might excite whatever that neurotransmitter ends up touching, whatever that other neuron is on the other end of it, or it may inhibit activity of that next neuron. Different neurons typically only have either excitatory or inhibitory neurotransmitters. It's extremely rare that you find one neuron with both types of neurotransmitters in it. So you'll have a neuron be classified as either excitatory or inhibitory. Neuroglia are the supporting actors for the neuron. They're helper cells that are unique to the central nervous system or unique to the nervous system, but don't really conduct electrical impulses. However, they do help the neuron do its electrical impulse receiving, generating, and transmitting thing while protecting it and making sure that its activity is shielded from any kind of outside influence. Astrocytes are named as such because when you look at them under a microscope, they look like stars. These are very highly branching, abundant, and versatile neuroglial cells. It has all of these branching processes. There's a process, there's a process, there's a process that clings to neurons, that clings to synaptic endings, that clings to blood vessels, specifically capillaries. And they do a variety of functions. They can provide physical structural support to these structures, to these entities, where they help keep them in place and keep them where they're supposed to be. They can act as a transmission medium between one type of cell and another, so between, say, capillaries and the neuron. And it also provides most of what's called the blood-brain barrier. So you can't get stuff from your blood directly into the neuron. If there's viruses in your blood, if there's toxins in your blood, if there's whatever, you don't want that just going to the neuron just easily, right? The astrocyte can serve as a mediator and help keep things out of the bloodstream that don't belong in the neuron and make sure that things are brought in from the bloodstream that the neuron does want. These regulate the chemical environment of neurons, and they especially help regulate leaked out potassium, and they help recycle neurotransmitters as well, so that you can hang on to the electrolytes and the ions you need to conduct action potentials easily and efficiently. And you don't have to go making any more neurotransmitters than necessary because you can recycle some of them. The astrocytes have a role in that. Microglial cells are also very highly branching. They tend to have more of an oblong cell shape, though, to them, as opposed to astrocytes, which are a little bit more round. These are specialized immune cells of the nervous system. These cells don't derive from the same embryological origins as the rest of the immune system, and they are completely unique to and only act within the central nervous system. Now, spoiler, there's a bunch of research coming out that suggests this might not be as true as we think it is, but for the purpose of testing, for the purpose of doing well on your TEAS test, whatever, this is right now the most supported fact, the most supported hypothesis, rather, that we have. These microglial cells will migrate towards neurons that it detects are injured, are old, have gone through apoptosis. If there's a bunch of cellular debris after something happened, the microglia will migrate towards whatever that is, clean up the dead stuff, and it will endocytose those. It'll phagocytose those components and deliver them outside of the nervous system if possible. Ependymal cells line the dorsal cavity to circulate cerebrospinal fluid. Specifically, they line the ventricles and circulate cerebrospinal fluid. So you have certain cells that will make cerebrospinal fluid. The ependymal cells have lots and lots and lots of cilia that helps make sure that that cerebrospinal fluid moves and circulates to all the parts of the brain inside and out that need cerebrospinal fluid, so that you don't just secrete cerebrospinal fluid in one place and then it just kind of pools up there. That's not helpful. You need it to circulate around, and the ependymal cells do that. Oligodendrocytes are less branching than astrocytes, and they make the myelin sheath. I've mentioned the myelin sheath before as something that you find on the axon. What it does is it helps shield the axon in a similar way that rubber casing around your cell phone charger protects the conducting copper wire underneath. It helps shield that axon from unintentional outside influences. It provides structural support. It provides a means of more quickly transmitting information. We'll talk about the mechanism of that later. And it insulates. Ultimately, it's an insulator for the neuron. Your oligodendrocytes have processes. So here's your oligodendrocyte. They'll often connect to the bloodstream too and help regulate molecules that are going to come in or out of an axon also. Mostly they make myelin. So they've got these processes that are going to reach out to an axon and wrap around the axon. And it'll do this in a few different places. So there's a process that wraps around here. Another one might have a process that wraps around here, etc. But it has these flat processes that coil around the axons. You have a process over here that's going to go to a different axon. And what it'll do is it'll stretch out like a sheet of plasma membrane, wrap it around an axon, and coil it up from the outside in until it surrounds the axon. As it does this, as it coils around it more and more and tighter, cytosol is going to get pushed out, organelles are going to get pushed out. What you really have is a double-layered plasma membrane and nothing else that coils around it. This is pretty impermeable to basically everything. So it's going to help regulate the internal environment of that axon pretty well. It'll help insulate the interior workings of the axon so that action potentials can be transmitted faster and with less interference as well. Because myelin is basically just made up of plasma membrane, it looks really white. So areas with lots of myelinated axons are called the white matter of the brain. You find a lot of white matter on the deep aspects of the brain, where you have different parts of the brain that need to talk to each other. It's this big internal network, so all of your cabling is going to be inside. And you find a lot of white matter on the outside of the spinal cord, because you need to be sending those nerves out to other places in the body. Those axons are going to be traveling outwards to other places. Gray matter is where you find cell bodies of neurons. You also find dendrites, non-myelinated fibers, a lot of glia. Things like that that don't have much myelin are going to create this sort of beigey appearance, or at least less white appearance, and that's referred to as gray matter. The peripheral nervous system has some similar cells, but they have their own, they're not exactly the same cells, and they have their own name. Astrocyte type cells that exist in the peripheral nervous system are called satellite cells. And the peripheral nervous system analog to oligodendrocytes that make myelin are called Schwann cells. And they wrap around their whole cell body around that axon, not just a single process. So you'll have a Schwann cell next to an axon. It'll wrap around the axon, and then one of the processes will go underneath the other one and wrap around the axon, and that'll keep going around and around and around. Similarly to the oligodendrocyte, that process is going to pull itself so tightly and so wound up so many times that there's not going to be room for cytosol or organelles or anything, and that axon is really just going to be covered in layers and layers and layers and layers and layers of just plain plasma membrane. This is really impermeable to electrical changes, ion movement, things like that. And it can make sure that what's going on the inside of the axon is only going on the inside of the axon.